Can somebody confirm you can hear me?
Hear you.
Great.
Yes,
Thank
I
you.
can hear you.
All right, should be seeing my screen now. So today is April 10th, and we are in the lab. Today we're going to be doing Lab 01, the ubiquity of microorganisms. You should remember that both Lab 00 and Lab 01, the worksheet must be turned in by 8 a.m. Friday, or you will be dropped from the class. Any question about that? Also, let me remind you, the environment quiz is due this Saturday, 11:59 p.m. at 4:12. And you need to read the infectious disease research project. It's a file on Canvas. You need to have that read by next week. We will be talking about the infectious disease project next week. Let me show you where that is. Go to the main web page for the class. Go down to week 2 lab, and there's the infectious disease research project. This is a 100-point research project, and I'll talk about it next week. And there's an example of the infectious disease report. All right, any questions about anything? Let me see if there's anything else I need to talk about. No. Oh, good. The lecture page finished. So that means my computer will speed up. All right. Lab Module 1, the ubiquity of microorganisms. To every lab, there will be a little bit of reading, sometimes a fair amount of reading. This lab only has a few pages of reading. Read about culture media in the textbook from page 157 to 162. And then read about obtaining impure cultures, a page from 162 to 163. There's some video clips in this lab. Your learning objectives for this lab are: 1, know that microorganisms are almost everywhere. 2, describe the purpose of culture media. 3, be able to describe colony abundance and colony morphology. And then be able to define the following terms. What's inoculation? What do we mean by ubiquity of microorganisms? What is a sterile, like a sterile environment, something that's sterile? What is isolation? What is incubation? What is in vitro, like in vitro growth? What is a culture media? And you should realize that with culture media, we have 3 types. There's a broth, also called the liquid broth, and that is a liquid culture media. And then there's an agar. That's where you add agar to the broth to get a solid media. And we have 2 types of agars in a test tube. We have a slant and a deep, and we'll talk a little bit about those. And then what's a fastidious organism? What's a non-selective media? What does a ubiquitous mean? And what is a colony? Any questions about any of these learning objectives? All right, microorganisms are organisms that are too small to be seen with the naked eye. Viruses and prions are included in this group, even though they aren't alive. And you could argue they're not organisms because they don't have cells. We'll also include helminths, worms, in this group and in our studies because although the adults are not microscopic, their infective stages are microscopic. The phrase ubiquity of microorganism refers to the concepts that microorganisms are essentially everywhere. Sterile means something is free of all living cells. I guess that doesn't necessarily include viruses, but when we say something is sterile, there are no living cells in whatever we said was sterile, like a bottle containing media. In this lab, we're going to investigate the abundance and diversity of microorganisms in common environments. We'll also learn some basic lab protocols. A culture medium, also called a growth medium, is a sterile laboratory preparation that contains a variety of nutrients that are required for growing microorganisms in vitro, meaning in a lab. How we get a sterile culture media in the lab is we make it and then we stick it in the autoclave. That's usually how we get sterile culture media. You could do something fancier, like carefully boil it in a microwave or boil it in a double boiler, something like that. But usually we put it in the autoclave, which, if you don't know what an autoclave is, it's sort of like a pressure cooker. Culture media may be liquid. We call these broths, and they're commonly put in a test tube. The reason being, if you were to put a broth in a petri dish, it'd be very easy to spill it and then contaminate it. Adding agar to a liquid medium results in a solid medium. You can add other things to a liquid media to make it solid, like you could add gelatin or starch, but usually if we have a solid media, we've added agar to it. Agar is a gelatinous substance extracted from seaweed, and we'll talk a little bit more about it later in the term. So here's a video you should watch on preparing a pour plate. Note how the culture media needs to be sterilized prior to its use. Why must a culture media be sterile prior to it being used? Can anyone answer that? Nobody's going to attempt? Well, if this culture media was not sterile before you put your one species of cells in it, you would then grow it up and it'll be contaminated by other things because it wasn't sterile to begin with. And then you would have multiple organisms in here, and the result could be thrown off by the multiple organisms you didn't intend to be in there. So we usually use sterile media to begin with so that we know what we're growing. Any question about any of that? All right. So this is an agar plate. Usually it's a solid media because liquid media would be too easy to spill out. But we put agar in usually into a liquid broth to make it solid. Usually the liquid broth is put in a test tube, but we can put solid media in a test tube, and this we call a slant, solid media in a slant, and this we call a deep, solid media, which is very similar to the liquid media. We just put it in when it's hot, and then it cools in that shape. Any question about any of that? All right. A general purpose media is non-selective in function, meaning it doesn't select for any organisms in particular. It's usually complex in composition, and we use it to support the growth of most non-fastidious organisms. So something like E. coli, we can grow it very easily in a general purpose media because E. coli is non-fastidious. There are three general purpose media we use all the time in microbiology. The first is the third down here, nutrient agar. All it has is some nutrients in it and agar to make it solid. And then there's tryptic soy agar, something similar. Nutrient agar has beef extract and tryptic soy, which is from soybeans in it. Tryptic soy agar only has um, tryptone, which is from soybeans in it. And then there's nutrient agar with yeast extract. It's nutrient agar with yeast extract added, so it's a little richer. All of these are general purpose media that we use to grow many different organisms. They're non-selective and they grow a lot of organisms. They will not grow a fastidious organism, though. We'll talk about fastidious organisms later in the term. A fastidious organism is one that does not easily grow. It will not grow on a general purpose media. You need to add specific nutrients to the media before a fastidious organism can grow. Any question about any of that? All right. Labeling. It's vital that Petri plates and tubes are correctly labeled prior to use. The following items must be added to your tube or your plate. You must include your name or your initials, the date, the type of medium it is, like NA for nutrient agar with yeast extract or NA for nutrient agar. You must put the inoculum, and that's what you're going to culture on the medium. If you know what it is, you just write E. coli. If it's something like organisms you collected from the air and then you're growing it on the plate, you just say air exposure. And then the incubation temperature is optional. You may include that, it's optional. So here we have a plate with the person's name, the organism, the date, the media, TSA, and then the temperature it's grown at, 37 degrees Celsius. Any question about any of that? Now, this is telling you she's right on the edge of the plate, because if you're right on the plate, it'll make it harder to see the microbes in the middle of the plate. As it is, writing on the edge will make it hard to see it right there, but usually we don't grow much on the very edge. Inoculation is the placement or transfer of microbial specimens into our sterile culture medium. And usually you put in only one species and then grow it. And then how you grow it is you incubate it. Incubation is the placement of the inoculated culture media in an appropriate physical environment for a prescribed period of time to allow the growth of microbes. We usually grow things in an incubator, and you can have complex incubators like here, showing you all the dials and controls so that you have an outer door shown. Actually, the outer door is shown there. This is the inner door. And um, all our incubators can control the heat, meaning the incubator warms it up to a certain temperature controlled by the knobs here. Usually we set our incubators at 30 degrees Celsius and 37 degrees Celsius, but you could switch it to whatever you wanted. We have put water in there, so all incubators can have a container of water that adds humidity. That's important to keep the plates as well as the tubes from drying out. So if you have your culture on a plate in a humidified incubator, that plate can survive for about 2 weeks before the organisms will begin dying. If you don't have the water and it's not humidified, the plate will only last for maybe 3 or 4 days. Any question about any of that? So all incubators can control the time. You just put it in, then pull it out. when the time is up, they also control the temperature and they can control humidity. Fancy incubators can also control the amount of oxygen in the incubator and the amount of carbon dioxide, such as shown here. All of our incubators in the microbiology lab at Clark College can only control the humidity, the temperature, and then the time, meaning ours are pretty simple. But if you have a complex one, you can control the amount of oxygen and the carbon dioxide being exposed to the microorganisms growing in the incubator. And then the time, just for however long you grow it in. All right. You should note that the agar plates should be incubated in the plate upside down. So when you're growing them, make this your plate, cells on here, the agar here, they should be put in the incubator upside down. The reason is that the moisture will come out of the agar, condense on the bottom of the lid, assuming you can see this. Yeah, you can see it, and form a droplet. And then what it'll do, it'll drop down on the plate, which contains your colonies. It'll mix up the colonies, which will disperse the cells, which could ruin the experiment. If it may not cause contamination, but it does increase the risk of contamination, because if the water runs down the edge of the plate and then comes into the plate, the edge is not sterile and it could contaminate the plate. But usually the water just drips down and it would not contaminate the plate. So don't tell me the reason why you incubate the plate is to prevent contamination. It does reduce the risk of contamination, but it generally will not be contaminated. It just will disperse all your cells so you won't have individual colonies growing on the plate. So all plates are grown in the incubator upside down. The reason being is that when the water now comes out of the agar, where is it going to condense? It'll condense on the top of the agar, and that will wick the water throughout the plate so it never forms a drop, which drops down. And even if it were to drop down, who cares? We're not growing any colonies on the lid. So we always grow our plates upside down in the incubator. Any question about any of that? All right. So when you're done, you take the plate out and you look at visible bumps on the agar surface. Each of these will be a colony where one cell may have grown into a million. You actually need about a million cells before you can see the colony with your human eye. So one colony, the smallest will be about a pinprick, smaller than the point of this pen, or up to the size of the point of that pen would be a pinprick, and that would be about a million colonies. Anything larger is more, and usually we have several million cells in a colony. You can have hundreds of millions of cells in a colony, and they all may be descendants of one cell, but it's not correct to say that a colony is descended from one cell, because a colony can be formed by one cell, two cells, or a clump of cells. And looking at the colony, you would not know if it was pure or not, because if it's formed by one cell, that colony is pure. But if it's formed by two cells or a clump of cells, the colony may not be pure. Any question about any of that? A pure colony is one that is form by only one cell because they all the cells will be the same species in the colony. If it's formed by a clump, you can easily see that there may be more than one species in the colony. All right, here we have a little video for you to watch on binary fission, where one cell grows into many. I guess I can show that. It's probably going to be quick. If it isn't quick, I'll just show you the start of it. Assuming it runs. We don't have a commercial. There we go. So that's two
Bacteria
cells.
reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
All right, here we have some colonies on a plate, and you can notice some of the colonies are very distinct, have clear edges, and then some of the colonies are fuzzy with hairy or non-distinct edges. These are fungal colonies that look hairy and non-distinct, and then something that's clear colony, clear edge, is a bacterial colony. Any question about any of that? We'll come back to this picture. So these are individual colonies on the plate. Like I said, some of them may be formed by more than one cell. Like these all look like they're the same species, but this one is much larger. That was probably formed by a clump. And then these are formed maybe by individual cells, or at least two cells. Colonies are not visible in a broth culture because all the cells will grow together. They will not form colonies in a broth culture. You can notice some different colony characteristics, different colony morphological variation, difference occurring between different species. For example, I pointed out that these are probably the same species. That's a different species. That's a different species. That's a different species. In fact, that's not even a bacteria, but all these are probably bacteria. That's a different species there. Little white colony. And these are orangish, yellowish colonies. And that's like a fried egg colony. Different species than that one. And that one's a different species too. You can tell these by the different colony morphologies, that these are different species. Colonies have different characteristics. They have a shape. You can have a circular colony, a rhizoid colony, a regular colony, a filamentous colony, and a spindle-shaped colony. They have an edge, that's the margin. If the edge is entire, a round circular colony has an entire edge. There's an undulate edge, which varies, a lobate edge and a curled edge. I would never ask you to tell me, is this margin undulate, lobate, or curled, because they're too similar to each other. But I would ask you, is it undulate, lobate, or curled versus entire? Meaning I can ask you those because these two, this group and this group differ. And then I won't ask you, is it rhizoid or filamentous? They're too similar, but I could ask you, is it this group or that group? Any question about any of that? Once again, the margin is the edge of the colony. The elevation is the elevation of the colony. To see the elevation, you literally have to take the lid off and then look at the edge of the colony, meaning from the side. It's very hard determining, is the colony flat, raised, convex, pulvinate, or umbonate when you're looking down straight at the colony. For example, is that colony flat, uh, elevated, pulvinate, umbonate? It's very hard to tell. This one, I can actually notice that it's lower on the edge and higher on the center. So this one I'm going to guess is convex.
Could you tell the difference whether or not it's convex or pulvinate, or is that going to
Pulvinate.
be
No,
something
I
similar
cannot.
to?
I'm not ever going to ask you, is it convex or pulvinate?
Okay, cool.
You just need to know that pulvinate is more extreme than convex, but they're the same shape. Umbonate is mountain shape. You can actually look at that from the side and determine an umbonate. Umbonate's actually a rare colony. Usually they're convex or pulvinate, raised or flat. The size of the colony, if it's smaller than the point of the pen or a pencil, it is punctiform. If it is about the same size as the point of the pen or the pencil, it's a small colony. And if it's bigger than the eraser or the pen edge of the pen, then it is a large colony. And in between the point of the pen or the pencil and then the eraser, that would be a moderate sized colony. Any question about any of that? The texture is whether the colony is smooth or rough. And for some reason, this one always throws students more than the other. All of these colonies here are smooth. The only one that looks rough is really that one. Let me take another colony. Here we go. Colony B is rough. It's hard to see here, but um, colony C would be smooth. Yep, I'm on it already. Never mind. My mouse back. Glistening or dull. If the colony is shiny, it's glistening. And if it isn't, it's dull. The pigmentation is whether the colony has color or not. If you can see any color, then it is pigmented. So these are all pigmented. These are non-pigmented. So that would be white, off-white, light gray, non-pigmented. Any question about that? Black is not considered a color, by the way, and neither is white. So color would be something like red, green, blue. And then the colony can be opaque, translucent, or transparent. A transparent colony looks like a drop of water. Very few colonies are transparent, and they're kind of hard to see. A translucent colony, you can look through it, sort of like slightly muddy water. I don't see any translucent here. Let me go down here. That colony is translucent. This region of this colony is translucent. Colony B is opaque. I don't know if that says that there. No, it doesn't. But colony B is opaque. Colony C is opaque. But this colony is translucent. There are no transparent colonies in this photograph. because it would look like a drop of water and you might not even photograph. All right, any question about the colony characteristics? Generally, if
Um,
you have questions about a colony characteristic, I will give you this table. You should know the common shapes like circular, irregular, punctiforms, a common shape. That's the only term you need to know. I think everybody knows small, moderate, large. Somebody had a question.
oh, I was just wondering for the quiz, would you show us like a figure six and then ask us like questions about the colony characteristics or
I wouldn't ask you questions about the colony characteristic, but I may ask you a question like, is it shiny or dull? Is it rough or smooth? Okay. I won't ask you elevation because I can't give you a petri dish and then you can look at it from its side. But I could ask you the colony shape, for example, is it circular, irregular, filamentous? All right, there's two terms you should know about colony. That's the abundance. That is simply the total number of colonies on the agar plate. This one's too many for me to count. This one, I'm only going to count this region here, but that would be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 in this region of the plate. So that would make this one maybe 30 to 40 colony abundance here. And it's very difficult to count the colonies in this region here and that region there. Any question about any of that? So the colony diversity would be for that plate 30 to 40. And if I were to ask you a question, what's the diversity? And you were to say something like 25, I would give it to you because that's close enough to 30 to 40. But if you say something like 20 or 10, I'll say, nope, that's not enough. So the abundance is simply the total number of colonies on the plate. The diversity is the number of different kinds of colonies on the plate. And there you got to look at the colony characteristics to decide what the colony diversity is. It's coming back to this plate. We already know there's one, there's two, there's three, there's four, there's five, there's six. Assuming that's black, that's seven. And then that's a little white one, which could be the same as that one. So we're going to say at least seven, maybe a few more. So let's say 7 to 9 would be the range. And anyone who gets close to that, like if you said 6, that would be okay. But if you say 4, I'll say 1, 2, 3, 4, 5, in which case 4 wouldn't be close enough. On the other hand, if you were to say 30, I'd say no, because the total number on this plate is only 30 to 40. Any question about any of that? So your guess must be close. It doesn't have to be exactly the same as my number, as long as it's close to my number. Any question about that? And diversity is much harder to tell than total number, because abundance, all you have to do is count the numbers. Now, admittedly, it's hard to count them here and here, but other than these two regions, it's pretty easy to count the colonies. You just need to count them. Diversity is much more difficult. You have to decide, are those two the same? I think they're different, but I'm not sure. And then these two, for example, those might be the same colony. This is just a smaller one than that one. And then I think all of these are the same colony, even though that's much bigger than these. I think just a clump form, that one. And these are bigger than these because these are isolated. These aren't isolated. That's clearly a different colony than these. Any question about any of that?
Yeah, actually, is it ever appropriate to give like a range with those numbers, or do you just want to be as exact as possible?
Um, a range is acceptable, assuming that your limits are within my limits, meaning you're close. But no, especially on this one, you should give a range because it's going to be very difficult to count the colonies here and here. So you can take a high number and a low number. Let's blow this up. So that looks like it's a colony there, 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 there. And so there's at least 4 there, but you could count that as more than 4 there. And there's 1, 2, 3, 4, 5, probably 6. 5 would be the low number. And then you could count more. 1, 2, 3, 4, 5, 6, 7, 8, 9, as high as 9 there. So on this one, I think a range would be appropriate.
Okay, thank you.
Or the abundance. And then diversity, it would be very difficult to have one number for the diversity. If you had isolated colonies, it would be very easy to count them, in which case everyone should have one number. But that's not isolated, so it's hard to count them. I think that's it for the discussion. Here's a real world example of abundance and diversity. So what is the abundance of cats in this picture? Come on, somebody give me an answer.
7.
7.
7.
Yes. And what's the diversity of cat species?
2.
2. Cheetahs and lions. Those are a male lion, a female lion, 2 lions and 5 cheetahs for a total of 7. Total abundance and diversity 2. So this one you should have the exact answers because it's pretty easy to count them.
So the sex doesn't matter for the diversity.
No, sex doesn't matter. It's just species.
Okay.
All right. So on the previous agar plate, is it possible to count the abundance and give me an exact number here? Same with the diversity, meaning give me an exact number. Is it possible to do that?
No.
No, you kind of have to give a range even here because, let me blow this up. Like right here, that's clearly one colony, but is that another colony right there, that ring? And is that a colony inside the colony? And those are clearly 2 different colonies, but that looks like that might be another colony. And then there might be another one there. And then at least one here, maybe several. And then one there and then one there. So right there, it'd be very difficult to count how many colonies are right there. So it'd not be possible to give me an exact number here. For this one, I might be able to tell you the exact number is 1, 2, 3, 4, 5 right there. That one's possible, but this one, not possible to get an exact number. All right, you should know the terms. There's a nice picture of somebody's hand. Put on the agar plate. And there's the answers to some questions. All right, let's go to the worksheet. You're not going to be directly doing this lab. Everything we do will be online, but you're expected to know the concepts and the terms of this lab. In lab module 1, ubiquity of microorganisms, the first procedure, we're looking at what is in the air you breathe. You have an agar plate. We expose it to the air for a period of time, which you got to look in the lab module, I guess, for that, because I'm not seeing it here, how long that was. Usually it's 15 or 20 minutes. Looks like you don't have that information. I cut out too much from the lab module. All right. Anyways, we then incubated the plate for 24 to 48 hours at 30 degrees C, and then pulled off the plate. And give me what's the colony abundance on this plate? And then what's the colony diversity? Put that answer there. And then what step would you do to reduce airborne contamination of a sterile plate while you are inoculating it? So let's pretend this is a plate here. Let's pretend this is my inoculating loop, which you can barely see. And while I'm inoculating it, what can I do to reduce airborne contamination falling into the plate while I am inoculating it? Don't give me an answer after I've inoculated it. Don't give me an answer before I've inoculated it, but while you're inoculating it. Give me the answer there. Diversity procedure 2. What gets on your arms when you put your arms on the lab bench? We have 2 plates, one that was streaked from the lab bench before you disinfected the lab bench. Use a cotton swab, touch the lab, put the cotton swab on the agar plate, incubate it, and then this is the cotton swab of the lab bench after you incubate it, after you disinfect the lab bench. Did table disinfection work? That's procedure 2A. Answer that. And then procedure 2B, what is the purpose of allowing the table disinfectant to air dry? Or why do we air dry the disinfectant? Answer that there. And the lab may not directly state that there. You could look that one up, or you can figure it out on your own. I think we did discuss it in a previous lab. Procedure 3, what is on your hands? So what we did was we had a student touch the plate with their fingers. There's one finger, one finger, one finger, another finger. So 4 fingers on the right hand, 4 fingers on their left hand. There's a little bit of their palm right there. I'm not sure why it's only 4 fingers. Maybe they were holding back their thumb. And this is before they wash their hands. And then we had the student wash their hands, dry their hands, and then touch the plate again with the clean hand. And you do need to have the hands dried because the first year or two I was doing this experiment, students were getting more microbes on their clean hands than on their dirty hands. And then I asked them, are you drying your hands? And they said, no, I didn't want to get microbes from the paper towels on my hands. And they had their hands wet when they were touching the agar, and the liquid whipped the bacteria from their hands onto the agar. And that's why they had more on their clean hands than on their dirty hands. Any question about any of that? So you do need to dry your hands, but there's the results after you wash your hands and dry your hands, and then before you wash your hands. And then does hand washing work? Give me an answer. And then 2, suggest one reason why hand washing did not result in the complete removal of microbes. So when we look here, there's less, but we do have microbes there. Hint, it may have something to do with the normal microbiota of your skin. And if you obtain an answer from research, give the brief citation where you got your information from. Okay? Procedure 4. When you kiss your significant other, what exactly are you giving him or her? What's on your lips and what's on your tongue? So we're talking about French kissing here. Here we have lips touching the plate, and there we have a tongue touching the plate. You should take a look and read the note I have above this picture here. And then procedure 3, I believe it is. Maybe it's procedure 4. Procedure 4A, which plate has higher colony abundance? The lip plate or the tongue plate? And then procedure 4B, why do you think this is the case, even though you might expect the abundance of both body parts, meaning your lips and your tongue, to be similar because they're physically next to each other, and you probably lick your lips? So what might cause the observed difference between the abundance of microbes on the lips and the abundance of microbes on the tongue? And to answer this question for full credit, you do need 2 answers. Any question about any of that? Meaning you need 2 reasons why one of the plates has higher colony abundance than the other plate.
I have a
This
quick
one,
question.
This one, yeah.
For the pictures of the plate, that the background on the tongue, like obviously the yellow colonies and like the reddish ones are there, but are those tiny, tiny little ones behind
Read the
that, or is
note.
that
Read the note above the picture. That would answer your question. All right. All right. 4C. Why was the tongue plate incubated at a higher temperature than the lip plate? We incubated this plate at 37 degrees Celsius and the lip plate at 30 degrees Celsius. Why did we grow the bacteria coming off the tongue at a higher temperature than the bacteria coming off the lips? Okay, that's 4C. Procedure 5. We're looking at bacteria we got from the soil. What we did was we took some soil, put it in a test tube, put it in water, took an inoculant of that water, and then streaked it out on the plate. And then for procedure 5A, what is the colony abundance of the soil plate? And then the question is it even possible to accurately answer this question? Please explain your answer. And then procedure 5B, is there greater abundance of the colonies from the soil compared to the colonies from the lips? Here. Now you note that most of the colonies from the lips are small, but there are a number of colonies in the soil which are also small. Like right here, we have a whole bunch of small colonies. This area here, this area there, that area there. So don't tell me that it's only we have big colonies on the soil and small colonies on the lips, because there are a bunch of small colonies on the soil. Almost all of this region here. These are more distinct than there. These are kind of blurred together. And those are a bunch of colonies there. So we do have small colonies in the soil. We also have a number of big colonies. Like there's a big colony, there's a big colony. But you have some big colonies from the lips too, like right there and there. Any question about procedure 5B? All right. Answer the questions to the worksheet. Make sure you turn it in by Friday, 8 a.m. because that's when it's due. And then if you don't turn it in by that time, you will get dropped from the class. Any questions about anything? All right, go ahead and work on the worksheet. I'll be here until 8 o'clock to help anyone with questions. But I will leave the class when the last student leaves. So if you have a question, make sure you ask it fairly soon. In
I
case
have a question
Mhm.
regarding the lecture. When is the slideshow gonna be posted and where?
The slideshow will be on the normal Canvas website. It will not be posted until I finish chapter 1.
Okay.
Okay,
Okay, thank you.
sure.
When we're citing sources for this lab, are we creating a page break and creating a whole new page for the works cited and citing it in MLA format or APA or whatever it is the format is that we're using?
To format it, you need to just state where it's from. So all you need to do is have the web address. If you want to do correct APA format, you give the name of the page, the author of the page, the last year the page was modified, if you can find that information, and then the address. But for this, all that's required is the web address of the page.
Okay, sweet, thank you.
All right. Um, if you have trouble getting to the lecture worksheet, you can get to it by clicking on the calendar and then clicking on the correct link in the on the calendar going to the worksheet. When you go directly from the web page, my Canvas web page, it looks like you can't add anything. There's no button to upload an answer. So you need to get to it from the calendar. If you have any questions, just email me. The two questions that people have asked is when they're submitting an answer, should they make their answers look different than the question? And yes, you should try to make your answer look different by either bolding the font, highlighting it, or using a different color. If you do use a different color, you should use a dark color so it'd be easy to read. But no, I'm not colorblind, other than in different shades of orange. I'm not colorblind, so any color will be fine. As long as it's a dark color so I can read it. All right, let me pause that. There we go. And that is, let me state that the links in the calendar to the Zoom meetings are not working. I'm not sure how they got there because I did not put them there. And one of them, the Microbiology 260 link, I don't name the class that. So let's just not use the links from the calendar. Use the link I emailed to you. I may put a correct link at the very start of the term, but the other links to the Zoom lesson in the calendar I'm going to remove.